Right. Super. Well, thanks everyone for coming. So today I'm going to tell you a story that we've been working on for a little while that was predominantly spearheaded by a couple of students. So Andrea Marsh, who is a former graduate student in the lab, and then Nellie Roberts, who is a current year 3 medical student. So I'm from Springfield, which is central part of the state. SIU School of Medicine is our home office. But what I'm going to tell you a little bit about today, let's see if this works, is rather than, much of what we've heard and much of what has been done has been looking at the nucleic acids and identifying whether or not the viral nucleic acid is there, so using a PCR technology or a related technology. But what we've been really more focused on is looking at antibodies. So, and the reason is because in medicine and in the medical field, people have been using antibodies both diagnostically and clinically for a very long time. But before we get to that, what I'd like to kind of introduce is just what are these viruses? So coronaviruses in general. So these have been around a long time. They've been around for decades. They're very common. They're single-stranded, positive-sense RNA viruses. And what does that mean? That means when they infect a cell, that their RNA looks just like our cellular RNA, and that it can be immediately translated into a protein or a gene product. But originally, there wasn't a lot done as far as recording of incidents or cases or deaths related to cases, and until the early 2000s. And that's when the very first, or the original version, just SARS-CoV, was identified. It was spanned across 29 countries. Wasn't, the numbers that we see recently, but there were about 8,100 cases, about 10%, a little under 10% death rate. But it didn't kind of continue on. It was kind of emerged and then disappeared. And then there was, about 10 years later, another coronavirus, pathogenic, the MERS strain or variant. 27 countries, fewer cases, but what you notice is that the death rate was about the same, so a higher percentage death rate for smaller number of cases. And then most recently, our friend SARS-CoV-2. So what I kind of wanted to depict with this particular timeline is that these viruses have, one, been around for a long time, but there's something very unique to this version that emerged in 2019. And it's represented by this little snow cone. And that's a protein that's found on the surface of this virus that's called spike. And why is spike relevant? It's not just a target for PCR-based assays, but it's what actually allows the virus to infect a very specific type of human cell. And it infects cells that express this particular receptor, and that receptor is called ACE2. And that's the formally called the angiotensin converting enzyme 2 receptor. It's found on epithelial cells of the lung, of the heart, of many other organs, but those two it expressed very high levels. And the one thing, just like other respiratory viruses and other viruses in general, I don't want to just focus on respiratory viruses, is that we get infected with viruses. Sometimes we have symptoms, sometimes we don't have symptoms. So you can be infected with a virus and you can not feel bad at all. And these are called asymptomatic infections. But regardless of whether you have symptoms or not, you can develop antibodies that are going to bind to specific proteins. Some of these proteins are going to be found on the surface. And in this particular case, so the body, B cells in particular, can produce antibodies that will bind to this surface protein, the spike protein, and actually block its interaction between the ACE2 receptor. So those are called neutralizing, meaning that they will prevent the virus from infecting its target cell. And that's what we're really interested in, is the body can produce antibodies. It does that naturally, as long as you have a healthy, functioning immune system, but it's going to be highly variable. And that's going to depend on a wide variety of things. And that's the beauty of the human population, is that we have a lot of variance. So what I'm going to tell you about today is what are we able to do in our lab in particular, and what many people are doing, is that we can detect whether or not these antibodies are present. We can actually quantify or determine the amount of those antibodies that are present. And it's that variance or that level of antibody. It doesn't just mean if you have a very high level of antibodies that means that all things are good. If you have a low level of antibody, it doesn't mean that all things are bad. So, because what you really want to know is what is the functionality of that antibody cocktail that's being produced by all these B cells. And so our assays couple these two things together. So we look at presence, quantity, and then functionality. So the overarching hypothesis is, and this is just not something new, is that neutralizing antibodies, they can prevent serious disease. They can also prevent death. And in fact, monoclonal antibodies were used as therapies initially and are still being used as therapies. And people are refining the monoclonal antibodies that are being developed. And that's eventually what we propose to do as well. But we think it's critical to be able to determine whether or not someone has antibodies and then the functionality of those antibodies. Because it can give some of our vulnerable populations and our first responders some level of protection, potentially. They can feel confident that they may be able to be protected if they have to encounter somebody that is very sick. So what we do in the lab is that we purify these two proteins. So I told you about the spike protein. So the spike protein is the major surface protein that is found on this particular version of coronavirus. And it has a functional portion that's called the receptor binding domain. So the ACE2 receptor is the target. So this full protein folds, and the receptor binding domain is what's going to poke out and then interact with this receptor. So we can purify both those versions. One thing that you'll notice is that the full-length spike is much bigger, right? These are not the size, for sure, but they're essentially, this is about 1/6 the size and amino acids of the full-length protein. So we can express those in cells, we can then purify them, and then we can apply them to plastic dishes. And once they're applied to plastic dishes, so we can use either the full-length version or the smaller portion, the RBD, the receptor binding portion, we can take serum from people, and this is just a blood product, so that can be separate the red blood cells, the white blood cells, and what you have left is serum. And then we can dilute that, and we do a series of threefold dilutions. So we start at 1:100, so that would be the first dilution that's prepared. Basically just taking 1 part of serum, putting it into 99 parts of saline or water. And then diluting that 3 times, and then 3 times again, and then 3 times again. So if you have antibodies that are present, that are able to interact with or bind to these purified proteins that have been now placed on these plastic dishes, they will be diluted out. And so I've tried to depict that with these upside down Ys that represent antibodies. But as you make these dilutions, you have fewer and fewer of these antibodies present. And where you get that reaction still occurring, then you're able to apply a numerical value. And that numerical value then, if you have a 1 to 8100 dilution that you've been able to go out to, and you still detect antibodies, then that's going to be a very high amount or high titer antibody. 1 to 100 is going to be a low amount because you can't detect it after you dilute it out further and further and further. And then our positive controls are, for these assays, are patients who we know have recovered from a COVID infection. So they have what's called convalescent sera or plasma. And then our negative control is just serum that we had in the lab from other studies that was pre-2019. So that was an assay to measure or to quantify the amount of antibody present. And then what we also do is we work with viruses for other purposes. So we're mostly interested in introducing new genes into cells using viruses, and we had a variety of viruses already available, and the ones that we work with the most are called retroviruses. also RNA viruses, but these viruses have the capacity to insert their cargo material into the chromosome of cells. And once that material is inserted into the cells, then it can report out. And so we use a reporter. So the big difference between the authentic CoV-2 virus and the viruses that we work with in the lab is that we don't need specialized facilities, so we don't need a BSL-3 or biosafety level 3 facility. We can work in a standard laboratory setting, a BSL-2 setting. And then our virus has a marker gene. And in our case, we use a marker gene that's called luciferase. And this is what makes fireflies glow. So, and it does this by emitting light. So when these viruses, now we can change their surface proteins so that they express spike. So everything else is different. They encode for a reporter. They're essentially a retrovirus, but they have this spike protein on their surface. So the only thing that is identical is spike. They still now interact with the ACE2 receptor, and that's the only way they can get into cells. And so our assay looks like this. So this is where we take our virus that we produce that now has spike protein, expresses this bioluminescent reporter called luciferase, and then we can apply it to cells that do or do not express the ACE2 receptor. So this shows specificity. And what you can see is that if the cells do not express the ACE2 receptor, that they're basically not infected. And if they do express the ACE2 receptor, then we have almost 100% infection. And that's measured by this light that's emitted from the virus. And that's depicted here. So no light in cells that don't express ACE2 and light in cells that do. So if we want to test the ability of serum to prevent this viral cellular interaction, then all we've got to do is take serum, mix it with the virus at an appropriate dilution. And we use a variety of dilutions, but we'll use a 1 to 50. So I told you about 1 to 100, 1 to 300. So we use a 1 to 50, 1 to 100, or 1 to 200 dilution in the case of the functional assay. But if those antibodies are present, then they can bind to the spike protein and block its interaction with ACE2, and then in this situation then we get no light. So just like with the cells that did not express the ACE2 receptor, now we can block that interaction so we get no light production. And so those are our two assays. So the clinical study that we took part in is we had 250 patients that were recruited between July and November of 2021. So this was prior to Omicron, which will become relevant later on. And then we collected information, so demographic information, medical information, whether the patient had been vaccinated or not. We also collected information on the type of vaccine they received, whether it was Moderna, Pfizer, or J&J. And then whether they had ever tested positive for COVID. We collected blood, banked serum, and then we had 50 of the samples from this 250 set that were blinded. So we didn't know anything about, they were just 01 through 050. Of that 50, we had almost equal representation of gender. About half the samples were patients that were under 55, half the samples were patients that were over 55, because we wanted to look later on at whether or not there was pre-existing conditions potentially impacting, where age being one of those factors. So for the biological study, we used the quantifiable antibody assay to look at both quantities of RBD and spike, and then the functional assay to look at how these things performed in the ability to prevent the infection of the virus. After all these assays were done with these 50 samples, then we unblinded the specimens, and we looked at how well or not our assays worked. So the first thing I want to draw your attention to is this is looking at the quantity of antibodies. So this is after we unblinded the samples. So we have a group here that was essentially vax negative, meaning unvaccinated, and never tested positive for COVID. The second group is a vaccination negative, so never been vaccinated, but had tested positive for COVID. Vaccinated only, no COVID infection, no positive COVID test, or someone who had been vaccinated and then tested COVID positive. So the first thing I'd like you to notice is that out of this group of 13, there were 7 individuals that had antibody levels to spike. So the only way that's possible is if they were infected with the virus, had no symptoms, and just didn't know at all, right? So they were asymptomatically infected. And we see a lot of variance. So we see that some people have very high antibody levels and some people have lower antibody levels. So it could be depending upon how productive the infection was, could be based on biology, but in any case, about half of the people that thought they were never, essentially knew they were never vaccinated, but thought they were never infected, had positive antibody levels. The next group is people who had recovered from a COVID infection. So they had tested positive for COVID and then recovered. And one thing you'll notice is that we no longer see the negative population, right? So these people are all testing positive for antibody levels, but they're kind of clustering down at the bottom. From our vaccinated group, we see that antibody levels were higher. Likely reason behind this is that the vaccine is based on spike, right? So the mRNA that's given to people is the spike encoding mRNA. So you've got all the antibodies being generated towards that particular antigen. Whereas someone who recovers from an infection, they're going to have a much more heterogeneous antibody production. And then we had folks, which is only a very small group, but they peak at the very top. So these 2 people that were tested, were vaccinated, and then had recovered from an infection, had very high levels of antibody. So now we took these 7 samples that were from these people that thought they'd never had COVID and knew they'd never been vaccinated, and then we looked at how they performed. Whoops. So our group at the bottom, which had the no antibodies detectable. So what we're looking at now is the percentage of neutralization. So the ability of the serum to block the infection of the cell by the virus, where 100% means completely blocked, and then 0 means not blocked at all. So we see that the group of individuals who were not vaccinated, not infected, they had no ability really to inhibit viral infection, whereas these people with super high titers, right, lots of antibodies, they were able to block viral infection very effectively. Someone with a little bit higher level of antibody, we see now they're a little bit better. Someone just under the highest level that we tested for, pretty good, right? So even out to the 1 to 100 dilution. So this is diluting 1 part of their serum into 99 parts of PBS. And then we're seeing still 100% blockage. And now we get into a little bit of a gray area. So these are people that are in the intermediate range, so 1 to 900. So not the highest, not the lowest, right in the middle. And what we saw is variable effectiveness of the antibodies to block the viral infection. And so you may wonder, why would that be? Why wouldn't the people that have the same titer have the same ability to block the viral infection? Well, if you look at this protein, as I mentioned very early on, so the receptor binding domain is only a small part of the full-length protein. So what you can have within the 1 to 900 group is that you can have antibodies that bind to the protein itself, but not in the working domain, not in the part that actually mediates the interaction of the surface protein with the cellular receptor. But some of them do. And so just based upon the way the antibody production is, that if we have kind of a homogeneous population of B cells that are producing those antibodies, then we're better off than if we have a population producing the other one. So out of the 50 samples, what we ended up identifying is that there were, there were those 29 out of 50 of the participants or in this blinded study, in these blinded samples, had greater than 75% inhibition of infection at the 1 to 50 dilution, which was the first dilution. And that the titers, based on the quantifiable data, were quite high, so 1 to 2,700 or more. And the spike titers were just a little bit lower. So as I mentioned, and that was probably because that's a smaller portion of the larger protein that we're screening for. So this was all well and good, but it was, again, we were working in the context of people who are vaccinated with mRNAs for the spike protein from the original version or original variant. So this was the Wuhan variant. It looks like this. So it has the receptor binding domain, then it has a single amino acid change. So we thought, let's look at something that emerged more recently. So after these samples were collected from our patients. So I told you July 2021 to November 2021. So since then, we've had a number of Omicron variants. And the Omicron has 28 different changes in the spike protein, 16 of which are in the functional part, the receptor binding domain. So now we wanted to test this serum from these patients that had high levels of antibodies against these two versions, so the original and this variant. One thing that we noticed in all of our studies, so as we were preparing viruses and just infecting ACE2 positive cells, is that the Omicron variants are somewhat more infectious. So even when we normalize for the same amount of virus, that they have a higher affinity or higher ability to bind to the ACE2 receptor that's expressed at least in this artificial system. So they're about 50% more infectious. But the one thing that we did note is that, so now we compared two patients, so each one of the red and purple bars is representative of an individual. So we have folks from the unvaccinated recovered group, the vaccinated only no infection group, or the double group. And then with varying, all the antibodies levels to spike, the full-length spike are very high, the RBD somewhat vary. But one thing is, once again, we're looking at percent neutralization, where 100% is going to be the best you can do, but 75% is our cutoff for saying that we believe that that's going to be a protective level, at least at this dilution threshold. And what we found is that even these sera from these patients is quite effective in neutralizing the functionality of now a version of the spike protein that has 28 different changes, 16 of which are within the working version. So what do we conclude from all this? So both these assays can work. They take time. It's a laboratory-based assay, but we can measure the amount of antibody. We can also determine the function of those antibodies. And it looks like, at least in our hands, using these blinded specimens, that the levels correlate with the ability to prevent infection or neutralize viral infection. So as far as the future directions, what do we want to do? We want to finish out the rest of these 29 samples, comparing them between the Wuhan and the Omicron emerging variant. And more importantly, though, is that what I would like to do is look more closely at this functional or working version of the receptor binding domain. So I told you it has 16 amino acid changes, but one thing that you'll notice is that there are regions that are not mutated. And so those regions are clearly required, conserved, for some functionality of the virus. So what if we make changes in those regions? So we can, based in the lab, we can make changes, we can change the structure of the spike protein, and then we can see, do we lose functionality of these antibodies based on changes made within those regions? We can also use those peptide sequences and develop monoclonal antibodies. Those monoclonal antibodies could then be tested for the ability to prevent infection. And we could use these assays, these same techniques, to be able to screen through those. We've already aligned these to many other viruses. Those regions are conserved across all the viral variants so far. So it suggests that there's something important to them, possibly, maybe not, but that's what we're going to test. So I thank you for your attention. I think we got a few minutes left, so be able to take any questions. Sure, go ahead.
Thank you for the presentation. My question may sound a bit clinical, but I want to find out the relationship between the neutralizing antibodies, I would say, to people having hypertension following COVID. Because I've seen people coming down with hypertension following COVID, or whilst they're being infected, they have this spike in their blood pressure. So is it because they're not really producing enough of the neutralizing antibodies, or the antibodies are maybe, attaching to a different site of the AC receptor? I don't know.
Good question. So I guess ultimately the question is this is during infection, right? So you're
Yes. So okay. So I'm just speaking out of experience of the people that, not my personal experience, from people around me.
okay.
So one of my colleagues, she's in the next hall. She had COVID and it spiked her blood pressure,
Mm-hmm.
and then she had to be placed on medication, and she's still on medication because of that. And I had another colleague, the same thing happened to him while he was
MAOI, like
yeah.
MAOI inhibitors, like antihypertensives.
Yes, yes. So the other one had a spike in his blood pressure while he was having COVID. But after he recovered, his blood pressure went back to normal. So I was just trying to, I know that, blood pressure has everything to do with AC receptors and everything, but I would think that people that are having COVID should, if they're having the neutralizing antibodies, it should actually make them be able to mount a good response so that they don't come down with hypertension. But
Yeah, I think I get where you're going.
Yeah, so I'm
So
just trying to think what could possibly be happening.
So I think in general, now if you think in general about viral infections, right? Viral infections result in inflammation. Inflammation as a whole, systemic inflammation, can cause high blood pressure or blood pressure to become elevated just due to the cytokines that are being produced. Now this is systemically. Endothelial cells in particular, which are mostly what our blood vessels are made of, as endothelial cells, do not express ACE2 receptor. It's cardiac muscle, mostly lung epithelium. It's more respiratory, right? But yeah, it takes you would have to administer those folks antibodies, right? Because this process of developing antibodies takes time. So, especially if you've never seen the pathogen before, if the body's never seen the pathogen. So, this is part of the humoral immune response. So it's B cell mediated. So you have to have this occurring over the period of, it's usually, a couple of weeks before, you have the innate immune system, which comes in, which is your neutrophils, your natural killer cells. And then the adaptive immune system comes in, which is the CD8 and the CD4 T cells. And the humoral system is kind of even more outside of that. So
it may not really have to do with the...
So it would have to be like convalescent serum that already had the antibodies, or an antibody cocktail that could be, that would neutralize. But I think it's most likely more related to just systemic inflammation. Go ahead.
Neutralizing antibodies, which is why it took us so long, years after the pandemic, to realize what is an effective protective GMT for these antibodies. So even in the ones who weren't infected and were asymptomatic, I don't know if is there a possibility to start correlating them if they get infected? Are they still less likely to ever have any symptoms? And if so, does it matter that they cannot neutralize the virus if they are fairly asymptomatic? Now, from a public health standpoint, they might still be able to transmit. So I think what I'm curious about is the correlation of neutralizing antibody titers with just having live, replicable virus in the system. How, if there's anything related to that even if it doesn't impact symptomatology, does it impact transmissibility? And there's a little data on how vaccinated people just don't shed virus as long, they don't shed virus as much, depending on the variant.
Right.
So I
Yeah, and I've got to say we pushed the envelope hard on these like infectious titers that we were using. So we were using 10 to the 7 infectious units per mil, which is like as an inoculum, that would be a lot. So, so I think as long as the antibodies can find the virion, then they should be able to. There's enough affinity, and you can select for super high affinity antibodies as well. So then you even enhance the possibility that you would have a very strong interaction between the antibody and the surface protein. And there's a lot of this, you know, these are glycoproteins, so there's a lot of it on the surface of the virus. Even though they're minuscule, it's mostly just all glycoprotein on the surface. But our assays or our experiments couldn't answer that question. I can only surmise.
Maybe for future directions, add that onto the list. I think we're just, again, it's just been a challenge, because the other challenge with the applicability of antibody data and making this translatable is that we don't know the duration is too variable. So you might be protective today and have this high titer, and some people, for them a week from now, they won't have a protective titer, and for a person too, a month from now, they won't have a protective titer. So unlike, say, hepatitis B or other titers that we measure, varicella, measles, that we're like, okay, you're protective, we don't have to check you for another couple of years.
Right.
It's hard. We don't have that duration of immunity, so the question is, how do you translate this into something that you could apply?
Yeah, and I think it would be doing those prospective longitudinal types of experiments, right? Is that this is from one draw that occurred in these people. I mean, they were, most of them, pretty far out from either the, you know, some of them didn't even know they were infected, right? These, so, but where we do have numbers, they were, you know, many weeks out from either being vaccinated or testing positive. So it was 36 to, you know, 45 weeks. So, yeah, go ahead, sorry.
No, that's okay. Just on your positives, these sound like they were early pandemic samples, so were they all infected before vaccination, I assume? Do you have any samples that were vaccinated first and then had breakthrough infection?
Yeah, those two were actually vaccinated first, breakthrough. And what we don't know is what was their level of antibody before, right? So were they in that, because even if you look at the people that were vaccinated, right, even though they kind of, the pyramid goes up so that you have the base going at the top, there still are people that don't have high titers. And that's common. You know, vaccinology is based upon aggregates. It's never based on individual dots. So we, you know, what we're trying to define, if we can, is to say, well, if you have this potential level, and we can monitor you over the long term, then it's possible that you can have some level of kind of confidence. You know, I don't want to say that you're protected, like you have sterilizing immunity for yourself, right? But I think you could maybe give people a level of confidence that you have you know, an 80% or statistically relevant chance of being protected in the environment that you're in. So, but I don't think we ever want to go to that point where we say, oh yeah, you're fine, because that's where you get into trouble. Go ahead.
Just, given the discussion about SHIELD, what do you think about the potential of looking for antibodies in a saliva sample rather than serum?
In saliva, we could try. Saliva historically is not a rich source of antibodies. So, you know, there's a lot of proteases, a lot of nuclease. I mean, it's to be able to use saliva for antibody discovery would be challenging. Still think we'd have to go to the blood draw. But once we do that, if we can identify a synthetic, then that's more the macromolecule type of system.
So when we make antibodies, we obviously make molecular antibodies for the same S protein, right?
So we make antibodies to peptide sequences.
Which are randomly presented by the THC. So you make different set of antibody for the same vaccination, I make different.
Exactly.
So have you captured, or is there a way to capture what is the variability antibodies that people with COVID, without COVID don't make it. Maybe this is a proposal for the.
That would take huge numbers of people, and there are people at Mount like Sinai that are looking at large populations for antibody heterogeneity, homogeneity, right, and seeing are there specific epitopes that are presented. The problem is that there's so much variation within antibody production within the human population that I think even if you had millions of participants, you're still going to say there's a lot of variation within antibody production within the human population.